Hello students, let us continue with the chapter Heredity and Evolution. In the previous video, I had explained to you about Mendel's experimental material and why he had selected garden pea or Pisum sativum as his experimental material. In this video, we shall be mainly focusing on mono-hybrid cross. We need to understand this in detail. Mendel took pea plants with different characteristics. A tall plant and a short plant produced progeny from them and calculated the percentage of tall or short progeny. In the first place, there were no halfway characteristics in the first generation, or we can call it F1 generation. F1 generation stands for first generation. In the F1 progeny or F1 generation, no medium height plants were found. All the plants were tall. This meant that only one of the parental traits was seen and there was no mixture of the two. So the question arises, were all the tall plants in F1 generation exactly same to the tall plants of the parent generation? We will be examining this in detail. Mendelian experiments test this by getting both parental plants and these F1 tall plants to reproduce by self-pollination. The progeny of the parental plants are, of course, all tall. However, in the second generation or F2, the progeny of F1 tall plants are not all tall. Instead, 1/4 of them are short. This indicates that both the tallness and the shortness traits were inherited in the F1 plants, but only the tallness trait was expressed. Thus, 2 copies of the trait are inherited in each sexually reproducing organism. These 2 may be identical or may be different depending on the parentage. A pattern of inheritance can be worked out with this assumption as shown in figure 9.3. Now students, let us understand figure 9.3. You can see that a tall plant which is represented as capital T is crossed with a short plant that is small t. When the tall and short plants are crossed with one another, the result, that is the F1 plants, are all tall plants and they are represented as capital T, small t. Now you might have noticed that in the parental plant, the tall is represented by capital T. That means both the alleles of the gene are the same. The short also is represented by small t. Here also both the alleles of the gene are same. But when we come to the F1, we see that tallness is represented by capital T, small t. Here, both the alleles are not same. Their tallness is represented by capital T and the other one is small t. But still the plant is tall because the characteristic tall is dominant over short. The short characteristic can be expressed only when both the alleles are recessive. If only both the alleles are recessive, the characteristic short will be expressed. But if along with the short a dominant characteristic is present, only the dominant will be expressed. This is the reason why in the F1 only the characteristic tall is expressed. Now the F1 plants are once again crossed or self-pollinated. That is capital T small t into capital T small t. When these are crossed, we get the F2 generation. In the F2 generation, we get 3 tall and 1 short. That is capital T, capital T, capital T, small t, capital T, small t and small t, small t. 1/4, you can observe that 1/4 of them are short and the rest are all tall. But all the tall are not similar. A few of them are hybrid tall and a few of them are pure tall. Now coming to the explanation of this, we need to understand that when we see 2 alleles that are same of the particular gene, that is for example capital T, capital T. Such a condition is known as homozygous, or for the case if small t and small t come together, it is known as homozygous. The explanation for this is having 2 identical alleles of a particular gene. When the 2 alleles are identical for a particular gene, it is known as, this condition is known as homozygous. Therefore capital T or small t is known as homozygous, while capital T small t is known as heterozygous. That means 2 different alleles of a particular gene. Now I hope now you have understood the difference between homozygous and heterozygous. Now coming to monohybrid cross. Let us understand this clearly. A monohybrid cross is a genetic mix between 2 individuals who have homozygous genotypes or genotypes that have completely dominant or completely recessive alleles, which result in the opposite phenotypes of a certain genetic trait. Now let us find the explanation for a monohybrid cross. Let us imagine that we take 2 pure breeding parents, that is capital T into small t. These are the parental plants and both are pure breeding, or we can say both are homozygous. When we cross a pure tall plant with a pure short plant, the gametes that we get are capital T, small t. When these gametes are crossed, we get the F1 generation. That is capital T, small t, capital T, small t, capital T, small t, capital T, small t. That means all the plants of the F1 generation are tall. But children, now you can observe that this F1 generation plants, though they are tall, they are not similar to the parental tall plant. The parental tall plant is pure breeding or homozygous, while the F1 plants are heterozygous. Now, these F1 plants are then self-pollinated. That means capital T, small t into capital T, small t. The gametes are capital T, small t, capital T, small t. When these are crossed, in the F2, the progeny that we get are capital T, capital T, capital T, small t, capital T, small t, small t, small t. So, capital T represents pure tall, capital T, small t, hybrid tall, again capital T, small t, hybrid tall, and small t is short. Now, we have to find out what is the genotypic ratio and what is the phenotypic ratio. Phenotypic ratio means when we look at a plant or when we see a particular organism, the look of it or the habitat of it is known as the phenotype. So, the phenotypic ratio here is 3:1. That means 3 tall plants is to 1 short. While genotypic means genetic difference. So, genotypic ratio here is 1:2:1. So, 1 means 1 pure tall is to 2 hybrid tall is to 1 pure short. I hope children you have understood the difference between phenotypic ratio and genotypic ratio. Phenotypic means how a plant or an organism is to look at, that is, and genotypic means genetically how it is different. So the phenotypic ratio is 3:1 while the genotypic ratio is 1:2:1. Now we have to find the percentage or in the phenotypic ratio. First of all, let us find out the percentage of tall plants in the phenotypic ratio. Now, when we take the phenotypic ratio, the ratio is 3:1. That means total number of plants are 4. So, to find out the percentage of tall plants, 3/4 into 100 will give 75%. While short plant, only 1/4 into 100 gives 25%. So, the phenotypic, according to the phenotypic ratio, the percentages are of tall plant is 75 and short is 25. Now let us analyze the genotypic ratio, the percentage of the genotypic ratio. Once again, the genotypic ratio is 1:2:1. So 2 plus 1, 3, 3 plus 1, 4. Again, total 4 plants. So 1/4 into 100 gives 25. Again, 2/4 into 100 gives 50. Again, 1/4 into 100 is 25. So, short plant is 25, hybrid tall is 50 and pure tall is 25. So, thus we have found out the percentages of the phenotypic ratio and the genotypic ratio. In the F1 generation, all the plants are tall. This means that only one parental trait is seen in the F1 plants. The F1 plants were then self-pollinated. Thus, in the F2 generation, not all the plants were tall, but 1/4 of them were short. This indicates both tall and short traits were inherited by the F1 plants, but only the tall was expressed. Why? Because tall was dominant over the short. Therefore, 2 copies of the trait are inherited in sexually reproducing organisms which can be identical. Example, or we can say which can be pure or homozygous or different, that is hybrid or heterozygous, depending upon the percentage. Now this is the explanation for the monohybrid cross. I hope all of you have understood the explanation for the monohybrid cross.